So, Dr. Marco Cristancho, he's a researcher at the Universidad de los Andes, and he's a leader of the Cabana project in Colombia. He's currently also the scientific director of the Center for Bioinformatics and Computational Biology. And for more than 23 years, he was a research scientist for the National Federation of Coffee Growers in Colombia. So he knows a lot about coffee. He's published over 30 papers, 3 books, many book chapters, and well, I think I'll let him introduce his talk. And
Gracias, Jan.
De nada.
Bueno, primero que todo, muchas gracias a Alfredo, a Celine, a todos los organizadores de este evento aquí en Irapuato. Es una maravilla realmente, como les he dicho en todos estos días, venir a Simvestab, tener esta joya en Latinoamérica. Es un privilegio estar aquí. Bueno, creo que ya muchos de mis... ¿Con cuál? Ya muchos de mis... I'm sorry, I have to move to English, right? So many of my previous speakers today, my colleagues, talk about many things that I'm going to talk today. So they did a good job to introduce my talk. So I'm going to talk about some application of genomics and bioinformatics in the fight against crop diseases in Latin America. And this is one of the reasons we decided to have food security as a major area of research for Cabana. So we actually included that the study of many of our crops in our workshops, in our research, in all of our activities that we did these 5 years, so almost 5 years that we conducted Cabana. Today, I'm going to refer to 3 examples, 3 particular examples of those diseases that threaten our production or crop production in Latin America. One is one of cacao, one of banana, and one of coffee. Cacao is a plant that it's original from the Americas. It seems that the origin is from the Amazon, but it moved thousands of years ago up here to Mexico. It seems that it was actually it was commercialized. It was produced somehow by some of the indigenous people that live in Mexico and Central America. So it moved all the way from the Amazon to here, to Mexico. Banana is this is a crop. This is a plant that is from Asia, but many of the producers, the main producers of the world, are here. And coffee, that is a plant that is original from Africa, but it adapted very well to the American continent, and most major producers are here in America. So the 3 diseases that I'm going to talk about are witch's broom, which is the main disease of cacao, Fusarium, TR4, which is the main disease of banana, and the coffee roast. They can cause losses up to these numbers. In the case of cacao, the witch's broom cause losses up to 60%, coffee roast up to 30%. And in the case of banana, we are getting crop losses of 100%. So the crop is completely destroyed by this disease here in Latin America right now. So it's a major threat right now for the banana production in Latin America. So witch's broom is caused by this fungus. It's classified into various biotypes, which is based on specificity, as is the case for most fungi diseases in plants. And one of these biotypes, the C biotype, infects a species of Theobroma, the cacao, and Erania in this group of plants. There's currently no effective control method available for witch's broom. In several cases, the plants have to be destroyed when the when the plant arrives, and there's not like any, once the plant is affected, there's not like any fungicide that you can apply to control the disease. It is currently limited only to South America. It hasn't moved to the major cacao growers in Africa. Ivory Coast is the major cacao producer, and so far they don't have witch's broom. So it is currently limited to South America, and in Central America only Panama and the Caribbean carry this disease. Those who have evolved with cocoa in its center of origin, and it was first recorded in the Brazilian Amazon in the 18th century. The second disease I'm going to mention today is Fusarium. It's caused by this forma speciale of Fusarium noxisporum. It is forma speciale cubensi, and is RT4, which is actually it's tier 4. Sorry, it's race 4, tropical race 4 of the fungus. So in fungal pathogens of plants, we got variants like the ones that we have been talking about all these years of COVID. In the case of fungi, most of them are called races of the pathogen. But it's a very similar concept to the one we managed for COVID, like the variants, Omicron, Delta, all these things. So these are the names that this disease received: Mal de Panama, Focube, tier 4, and Marchites del Banano. There has been some controversy in the last few months about the classification of this Fusarium. And I'm going to show you, I'm going to talk about it later on when I talk about the genomics of the fungus. Okay. It causes leaves to turn yellow and wilt, and vascular discoloration, which is only visible if the pseudostem is cut open. So you will see the plant like this, but you will only see all this discoloration of the vascular system when you cut the stem of the tree. It's most commonly associated with Cavendish varieties of banana, but can attack other varieties. But in here I have to mention that one of the problems with this disease, and actually with the banana crop, is that it's been a monoculture most of the time, that it's been a commercial product. Before Cavendish, we had something that it was called Gros Michel a few decades ago. And more than 90% of the crops, of the banana crops of the world, were cultivated with Gros Michel. That Gros Michel was affected by one strain of Fusarium. So the breeders could find a replacement for that Gros Michel, and it was Cavendish. Cavendish was resistant to the Fusarium variety, the Fusarium variant that we had at the time, the Fusarium race that we had at the time. And everybody started cultivating all over the world Cavendish. And right now, it stands for over 99% of the commercial banana of the world. It's only Cavendish. Well, Cavendish is now susceptible to the new mutations of tier 4. And as I showed, and as I showed you, it destroyed completely the banana production. So basically, you see all this in the media and in their main media and also in the scientific media, that there's a pandemic destroying the banana fruit everywhere. So let's have a look at how this pathogen has moved through the years. In the 70s, it was first found in Asia. In 2013, it was found in Africa. And in just 3 years ago, was identified in Latin America. The first country that identified the pathogen here in this year was in Colombia. And in 2021 was identified in Peru. Those are the only 2 countries where it has been identified so far. Northern countries have said that they have the disease yet. Of course, there's a local emergency, regional emergency, because so far the situation in Colombia is pretty bad, where the disease has been detected. Complete crops have been destroyed by the disease. So to mention that the reports here have been done by what we've been talking about all these 2 days, and it's basically the use of sequencing using genomics bioinformatics to identify the current variant, the current race of the pathogen in Colombia and in Peru. This has been done. I wanted to show you that here in Mexico, I found this action plan for the surveillance and management control of the disease. So you got like a booklet here in Mexico for doing that in case it is detected. The last disease I want to mention today is coffee rust. That's the one I have worked the most during my career. And this one, I would say most plant diseases are pretty much related to climate and what the climate is. And in the case of coffee, we could actually demonstrate that, as I'm going to show you. So as with most crops that I have mentioned today, cacao, banana, and all tropical crops, this one is also mainly grown in tropical areas of the world. Here in Latin America, we got some of the largest producers. Brazil is the largest producer of the world. Colombia is third. It was replaced by Vietnam, which is the second producer now. You got production in Africa and large producers in Asia as well. How the rust moved in all these coffee producing areas. I'm just going to mention when it did arrive in America. It arrived in Brazil in 1970. And there are some different theories of how it arrived. There's a theory that it was even published in Nature a few years ago, because we think that men brought the disease in a plant or anywhere, in clothes, could be brought anywhere. But somebody had this idea that it came through sandstorms that came from Africa, and it had all kind of proofs that that was the way that the disease moved. So we don't know how it moved, but anyway, it arrived here in 1970. It started rapidly moving through all the continent. In Colombia was curiously one of the last countries to arrive in 1982, because we had a lot of different mechanisms to try to control the arrival of the disease, but still we couldn't manage to stop it. It arrived here in 1982. So now it's everywhere in the world. The last places where it arrived was in Hawaii, which is the only place. Well, no, it's not the only place. It's one of the places that is grown in the United States. Now coffee is also grown in California, actually. So I'm going to mention a couple of things quickly before I move to the part of genomics and the other stuff. We had a recent outbreak in 2007 all over Latin America. What happened was, I showed you when the disease arrived, and after the arrival, we had bad epidemics of rust in most places, but it lasted for 5-6 years. After that, it sort of slowed down, and it wasn't so bad. For at about 1990, 1995, the disease wasn't as bad as when it started. It wasn't an epidemic. So for almost 30 years, we didn't have any epidemics of rust. We in Colombia and in several countries in Latin America, the way we combated the disease was with resistant plants. So the plants was the solution for this disease. So for almost 30 years, we combated with resistant plants, but still some growers still had susceptible plants for many reasons. And in those 30 years, many coffee growers changed resistant plants for susceptible plants because they thought the disease had disappeared. We didn't have any problem. It wasn't a problem until it came 2007. And I got a call from people in Colombia, and people started calling from all over the places, from all over Latin America, saying, you gotta come here and see what's going on, because we didn't have. Let me see if I got here. No, got it later. We hadn't looked. I didn't. I wasn't a witness of coffee roast epidemics. When I arrived to work with coffee, the epidemics had already passed. So all I could see was just some spots of the disease, but not the plant being in such a bad stage as when you have an epidemic. So people started calling and saying, look, we have a really different scenario. And we realized we were in a really bad stage. So most people say, well, we have a super race because we didn't have the disease and suddenly it just exploded. We had to prove if it was a super race or it was something else. And just to make a long story short, we proved that at this time, 2007, 2008, the race that was in the coffee fields in Colombia and other places in Latin America was the same race that we had 20 years ago. What happened was, in all these years, we had climate change. We had almost 3 years of continuous rain in several places in Latin America, and specifically in Colombia. So it was related to that we had these new epidemics. And also to the fact that people have moved from resistant plants to susceptible plants. Okay, so it was these two conditions that were really different from what we had before. Okay, we published that and with several colleagues from Costa Rica, Nicaragua, Guatemala, from several places. I'm going to show you finally the molecular studies of these diseases. So in the case of the witch's broom, this is the size of the genome. It's a regular size for a fungal genome. Most fungal genomes are in the order of 30 megabase pairs to 100 megabase pairs. The sequencing was done at Unicam in Brazil. And this genome has 8 chromosomes. For most fungal pathogens, one of the problems that we have is we know very little about the genetics. Probably it's something that we also have discussed with Sofian about this genomic surveillance. How we need to know more about genetics, not just genomics, because we know very little about the genetics of most of these pathogens. We don't know how many chromosomes there are there for most of them. We don't know if they are diploid or haploid or what ploidy condition they have. Some of them have several nuclei. So it is a challenge to work with these guys because it's very little we know about the genetics. I'm going to show some of the solutions that have been proposed to deal with these diseases, especially for witch's broom and for banana fusarium. This is one of the solutions that people are saying is probably the only tool we have right now to combat the disease, and it's gene editing of the plant. So it's something that we have to discuss, but I think right now it's probably the only technological solution to combat this problem. In the case of Fusarium, the genome size is this one. It's a little bit larger. We have sequenced 59 genomes, 23 from the tier 4 race. and 7 from South America, 3 from Colombia, and 4 from Peru. Those 7 have been done just recently, and the people from all these organizations have used Oxford Nanopore for the sequencing of these isolates of the pathogen. So it's been a nice collaboration between colleagues from Peru and Colombia to do the sequencing of this major pathogen. So focus strains are also divided into races according to their host specificity. The same for cacao, which is broom, the same for coffee roast. But also the same for the other two. We don't know which variants and factors underlie this interaction. So genomics should help us do that, should help us understand what are the variants and virulence factors, the ones that Sophie mentioned about in his talk, which underlie these interactions with the host. We know that this race 4 harbors 361 homologs, which are candidates of virulence genes in this pathogen, but it's under study right now. These genes. So again, for banana Fusarium, it's been suggested that gene editing or CRISPR or these technologies can be the solution. And from what I can see, is probably the only solution. Banana, especially, is a crop very, very difficult to breed to do normal breeding. So you just got to rely on natural mutants. So for a crop like this, I think this is going to be the only solution once you have this disease in place. Something that I wanted to mention quickly is this paper that recently was published, and it's something that it was mentioned today. Because these are soil-borne diseases, we could do some soil management to try to manage the diseases. So it's something that is being suggested that we should consider right now to do. Finally, I'm going to talk quickly about the Millet basatrix. There have been 3 rounds of sequencing. We did sequence the first isolates in 2015 using the old 454 technology that is not in the market anymore. In 2015 and 19, sorry, some people from Embrapa in Brazil used PacBio technology to sequence the genome. However, this genome is not a good assembly. None of these 2 are good assemblies. So we decided to resequence again and use the race 2, which is the prevalent race in Latin American crops, and use sequence also Brazilian and Peruvian isolates with the Cabana resources we have, the Cabana funding we have for that. The coffee roast genome is much larger than the other guys. You remember that the other guys were about 30 megabase pairs, and the other one 50 megabase pairs. This is more than 10 times that size. This is even larger than several plant genomes. So it is a monster in the fungal kingdom with this genome size. So it is a huge challenge to actually assemble and study this genome. For this, we sequenced race 2. The sequencing was done here at Simvestab. We used PacBio at this depth of sequencing, 160, Illumina 100. We sequenced 5 isolates from Brazil using Illumina, and 1 isolate from Peru using Oxford Nanopore. That part of that is already on ENA, but we've been discussing with Guy and some of you how to deal with the submission of this data, because we got over 200 gigabytes of data. So something I wanted to mention is that from this coffee condition we had in 2007 in Colombia, we moved to a much better condition in 2019. We did prove that this was caused by climate change, by climate conditions, rather than a super race. Nowadays, from this year until now, 2022, we got a new epidemic. It seems that is growing right now in Colombia and other places in Latin America. And this time we think that is related to climate, but we might have a new race. That seems to be the case now. So what's next? We need to assemble and annotate all this sequence data. And in the case of the coffee roast, we got over 200 gigabytes of data. That's a major challenge. We need some computer power. For this, we did something that, or we are doing something that Sophie mentioned, and it's a kind of crowdsourcing that we are trying to get people from all over the world, from not just Cabana partners, but from all over the world to try to do this work together of assembling all this kind of data, all this amount of data that we have. And then do targeted sequencing. We've been talking with Jan and some other friends about how to develop diagnostics and surveillance with this targeted sequencing. There's a technology called MARPL that we could perhaps use for these pathogens. And lastly, Ian likes a lot the what if, so I had to bring a what if. What if we continue working as a Cabana network and probably enlarge this network? That's our major goal right now. And we have to continue coordinating efforts to solve LATAM crop biotic and abiotic stress. As you saw, all these pathogens have been combated by Latin American institutions with international collaboration. But we have to try to bring our own solution to our problems. And let's keep the good momentum of having all these people working together. So announcements to CAD, Ian, PIF at EBI, and all these people who have been involved in all Latin American countries in this research. Thank you very much for your attention.
Thank you very much, Marco. So we have time for one or two questions, I think. Any questions? No. I have maybe a comment. I'm surprised that it's such an important pathogen, and the genomes that are out there are not even of very good quality, let's say. And how come is that, you think?
I don't know. They did PacBio, which usually gets you a good genome. They didn't complement with Illumina because sometimes, especially in the early stages of PacBio, you had to have like some Illumina data as well to actually do a good assembly. So probably that will happen, and they don't think they had the funds to do more sequencing, and they just went ahead and published a genome which we thought was going to be much better, but the improvement from the 454 genome wasn't as good as we expected. So I think that's the reason. Now we got PacBio and Illumina, so it should be a really good genome.
Good opportunity to contribute to that field of science. Ian, I think you have a...
Thanks, Marco. So all of these crops are major cash crops for Latin America, sure, and indeed different parts of the world. So is there not an opportunity here for more engagement with industry? And could this not be a low-hanging fruit for engagement with governments? I mean, this is a huge revenue for local governments and also for industry. And yet somehow, even in Cabana, we haven't really been able to engage with industry.
Yeah, that's a great question. And yeah, it's true. That's the reality. But in fact, what happens is because of the industry interest, the commercial interest, the cocoa genome was first sequenced by Mars, and they didn't publish the sequence. The coffee genome was sequenced by Nestlé, and they didn't publish the sequence. And so it's great when industry gets along with this because they got the money, they got the interest, they know that they can use it, but they don't want to share anything. It's pretty hard to actually commit with them so it's there's a lot of data of coffee, cacao. I don't know too much about banana, but probably. Yeah, but still, for them, they like to keep a lot of information for themselves and just work with some partners. They like to work. They don't like to make like large networks of researchers doing doing research in the crops. They just managed to work. For example, Nestlé does work with Colombia. They do work with Brazil, but they don't do work like tripartite. Brazil, Colombia, Nestlé. It won't happen. It will just happen in this partnership of two. So that is my experience with coffee. I don't know in cacao. They tried to do a large network, but I think it didn't work from what I could see. That it was just them working with few people, the people they wanted. Yeah.
Okay. Thank you very much. I think in the interest of time, we will continue.